Paris lab, and we're still studying dengue. So my project this summer is developing a neutralization assay to characterize the antibody response to dengue virus in humans. And so, again, adding to the background that Louis was saying, dengue is a mosquito-borne virus, and it does lead to severe disease, but it's still unclear why some individuals don't have severe disease and some do progress to shock or very strong fever. And it's very important to note that there are no vaccines or dengue-specific treatments, and so that is a little bit of a problem. Again, looking at this map, considering that about 400 million people are infected every year and 3 billion are at risk. So a vaccine is kind of, or some kind of treatment is very, very good, would be very good. And so again, just a little background on vaccines. So if you take a man and you vaccinate him, you basically give him a sublethal infection. That's kind of the theoretical aspect of the vaccine. And so they build a response, they build a little bit of an antibody response such that once they clear the initial infection, then later on they will be protected. So anytime they get infected again, they'll be able to fight. And so last year there was actually a very big vaccine trial that failed. And what happened was they were vaccinating patients and they were collecting the serum which has all the antibody, and they were testing the antibodies. So they tested the antibodies to see if the vaccine was working. And they thought that their vaccine was working. Based on their test, they said, okay, yeah, these patients should not be infected. But in clinical trials, this vaccine failed. Patients were still getting infected. So that's where the project that I'm working on comes in. We believe that the test that they were using was faulty. It wasn't good. And so we are developing a new test that we believe would be more predictive of what will actually occur in humans. And again, a little bit more about antibodies. So that little Y structure right there is a cartoon antibody, and the top half, the fork of the Y, is the business end. That's the part that targets that actually where the antibody targets. And the bottom is the constant region, also known as the FC region, and that stays constant with all the antibodies. So the top half can vary based on the target, but the bottom half usually stays the same. And this is so that the cells can come and pick up the antibodies once they bound. And there's a very important concept that Louis touched upon, but I'll just give a little simplified graphical model of neutralization. So you have a viral particle that's kind of the colored circle in the middle, and it's completely coated with antibodies. So that particle is said to be neutralized. And in contrast, you can have non-neutralized particles. So antibodies can be non-neutralizing for various reasons. You can just not have enough of them. So if it takes like 10 antibodies to neutralize a particle and you only have 5, then it's not going to neutralize completely. Or you can have like a different antibody, like this blue one, which isn't really specific. And so it doesn't bind very well, and thus it doesn't neutralize. So antibody-dependent enhancement is kind of a theory as to why some individuals actually progress to severe disease and why some don't. So if you take, this is kind of a cell, this double layer marks the cell membrane, and cells express surface proteins, and the natural dengue infection is the virus attaches to one of these surface proteins called DC sign, and the cell kind of eats the virus, and it goes into an endosome. And so the infection occurs when the virus fuses with the endosome and then releases its RNA, and then the cell is infected. So if you take a neutralized virus, again, a virus that's covered with antibodies, that can also be uptaken by the cell, but this time it's through a different receptor called the FC receptor. So this actually binds to the bottom end of the antibody from the constant region. And then this is taken up by the cell. So, but this time the virus can't bind to the endosome and cannot infect because it's being neutralized, it's being covered, it's kind of protected. And now, consider the case when you have a non-completely neutralized viral particle. So the antibodies are kind of only partially covering it. So the virus can still be taken up by the cell through the FC receptor, but this time the antibodies can't prevent the virus from binding to the endosome and releasing its RNA and infecting the cell. So you can see that if you have a certain class of antibodies, that they provide another mechanism for viral entry and for viral infection. So you can see that it's very, very important to characterize the antibody response that is generated by vaccines or by infections, because you can either get neutralization or you can get enhancement. And the assay that they were using in the previous vaccine trial couldn't cover this. And so they were using a test, and they were using the cell line. So a cell line is basically a clone of multiple cells. So you take one cell and you have like 5 million or billion or however many you want of these cells. And the cells that they were using in this vaccine trial was the so-called Vero cell. This is a monkey kidney cell line. And it's not a natural target for dengue. In no natural instance would a monkey kidney ever be infected with dengue. So they don't, not only that, they don't actually express Fc receptors or this DC sign surface protein. So if you remember from the previous slide, those are very important to measure the dengue virus infection and the antibody infection. So the whole point of the project that I'm working on is to switch over to this new cell line, the U927 DC sign cell line, which is a human monocytic cell line. Basically, it's a white blood cell, and it is a natural target for dengue. And it does express, I mean, these little squigglies are what I call the Fc receptors in the DC sign, but it does express these. So it can measure the natural infection, and it can also measure AE. It has the potential to do both. And so this is the test that they were using. It's called a neutralization assay. And so the basic test is where you have cells, like a layer of cells, and you come in with your virus. And the virus will infect the cells. And then we come in after the infection, and we stain the cells that are infected. So the cells that are infected turn blue, as you can see here. And so the more blue cells there are, the more infection there is. And we can count the number of cells, and we can use that to make measurements and compare. So just to give you a little idea what that looks like, you can do these tests in a 96-well plate. It's just kind of a plate like this big. And you can do basically 96 experiments in this plate. And you can see here the blue cells that are infected versus the kind of grayish ones. I don't know if you can see by the monitor, but those are the ones that are not infected. So there's a clear distinction between the two. Now, to test the antibody response, you just add a kind of a step in before. You mix the virus with the antibodies, and you kind of let them incubate and soak and mix and stuff like that. And then you expose the virus to the cells, like you did before. But this time, some of the virus particles are neutralized, so they cannot infect. And some of the virus particles will not be neutralized, and some will actually be partially neutralized. There's kind of a little bit of schematic. So again, after the infection occurs, we come in and we stain the cells. And so you can see that there should be a reduction in the amount of infection. As you can see, some of the particles are pulled out of the infectious state because they are neutralized. So if you have good antibodies, or if you have a lot of antibodies, you should see less infection, which means less blue spots, or fewer blue spots. So I'm going to get into a little bit of data. So here's some curves and stuff. Okay. So this is a neutralization curve. And so I'm just going to explain the axis first. So the y-axis is the relative infection. So you can think of this a little bit like the percent of cells that are infected. So the red dotted line that's kind of going across the middle, that's the baseline infection. That's what I'm calling the baseline infection. So that's basically virus with cells, so no antibodies. So the virus will infect, let's just say 10% of the cells. We call that the baseline. So now in the y-axis, we're talking antibody dilutions. So if you go down the y-axis, you get fewer antibodies. So if you look at the 4 viral particles at the top, it kind of gives you a graphical depiction of what's going on. So at the highest, or at the lowest dilution, where you have the highest amount of antibody, you have the highest amount of neutralization. So that viral particle is essentially neutralized. And so you can see that it's actually much, much lower than the baseline. And so it can, that's essentially neutralized. So you see, so basically in the assay you would see fewer spots. For example, if in the baseline you would see 10 infected cells, here you would see maybe 1 or 2. And then as you decrease the amount of antibodies, you can see that the infection actually rises and then comes up to baseline. So you can see at the very end that one poor little antibody is trying to neutralize the entire virus, and it's not very successful. And it just returns to baseline. So this is one neutralization curve from DV1 serotype. Lewis was mentioning the serotypes. Essentially, they're kind of like brothers. They're related, but they're kind of different. But yeah, they're genetically related, but a little different. And then this is another neutralization curve, and you can see it follows the same pattern. So we have the most antibodies. They're actually, it's being neutralized, so you see less infection. And then as you decrease the amount of antibody, as you kind of pull it away, you see more and more infection. And so this is a nice neutralization curve. Now I'm going to show you something really crazy. Okay, so this is actually the enhancement curve. This is what I was talking about before with the ADE. So it kind of starts out normal. So when you have a lot of antibody, you can see that the virus is being neutralized and you see reduction in infection. So it goes, and then suddenly you see a 6-fold spike in infection. So we're using the ADE model to explain this spike in infection, this dramatic increase. So if you see, I depicted that as the kind of the blue antibodies just for ease. But you can see that at a certain concentration of antibodies, you can actually get enhanced infection. And so this is very, very important that we're seeing this in our assay. Because in the previous assay, the one that was used in the vaccine trial, they would never be able to see this because they weren't expressing Fc gamma, Fc receptors. So they would never be able to see enhancement. So they could not even predict if individuals would get enhanced disease or not. And so that's kind of a major fault in the assay, and that's something that we're trying to overcome in developing this new assay. So just to briefly summarize, so again, it's not exactly clear why some individuals present with severe disease and why some individuals seem to be protected. But we believe that the antibody response is a major part of that. And so current tests that test the antibody response, they don't correlate with disease outcome. That was shown in this vaccine trial, where they predicted that all these patients would be protected, but then they got infected anyway. And so our test that we're developing, it captures both the neutralizing and the enhancing properties of antibodies. And so we think that this would be a much better assay, a much better platform to test for vaccine candidates and even for as Lewis was saying, like heterotypic infection, so individuals that are infected with a different serotype after their primary infection. So I would just like to thank the Harris Lab for running a very good work environment. The PI Eva Harris is amazing, and then I would like to especially thank my undergraduate, postdoc, he's an amazing person, and then Angela's also helping me a lot, and Alexandra's the amazing graduate researcher is very helpful. I'd like to thank Leah for organizing the SURF and for Nathan for helping me with my presentation. It was a little bit discombobulated earlier. And then I would like to thank the Rose Hills Foundation for providing the funding for this project. Thank you.